Our next speaker is Julie, and engineering transcription circuits to elicit new phenotypes in E. coli.
Thank you very much, Professor Silva, and good afternoon, everybody. It's my great pleasure to give a talk to you. I will talk about a powerful tool for engineering regulatory transcription circuits to induce the desired phenotype in E. coli. To start with, I will describe this research background. After complete genome sequencing of the diverse species, functional and comparative genome analysis can provide us numerous genome information. The next goal is to engineer the desired phenotype and functions at will. Also, the engineering of the desired phenotypes in organisms of interest is a major goal in synthetic biology towards engineering the complex biological systems and understanding their natural counterparts. But for phenotype engineering, the conventional approach, such as mutagenesis and evolution and relational engineering approach, can simply modify single gene or sequentially multi-genes. So, for the phenotype engineering, we need whole genome engineering approach to allow multiple simultaneous gene expression modifications. For this, transcription factor which thoroughly regulates phenotypes in the complex genetic regulatory network is a good answer. But innate transcription factor regulates just their target genes and consequently determines their specific phenotypes. So, for the desired phenotypes, a novel artificial transcription factor, ATF, to deconstruct innate gene regulatory network is required. So, in this research, we have developed a novel artificial transcription factors that are capable of reprogramming gene transcription in E. coli, and using the novel ATF, we induced various phenotype changes in E. coli. Once that was done, the genes associated with the selected phenotypes were characterized. To do this, the first step involved development of a novel ATF. We used the zinc finger proteins, which recognize a specific 3 base pair DNA sequence as a DNA binding domain, and as an effector domain, which regulates transcriptions by interacting with RNA polymerase. An E. coli catabolized regulatory protein, CRP, was used. CRP is a transcription factor with dual activities as activator and repressor in E. coli. Also, in order to select the effective effector domain, we engineered CRP proteins and constructed 4 kinds of the CRP derivatives. In short, we engineered CRP derivatives by deleting their own DNA binding domains and by including their activating domains. And then, to confirm their transcriptional activities as an effector domains, they were linked to a three zinc finger proteins that bind a specific 9 base pair sequence. And then we examined their transcriptional activities, activation, and repression. So, as a result, among those CRP derivatives, CRPD1 showed both strong activation and repression. So, using the CRPD1 effector domains, we have constructed ATF. And using a zinc finger DNA binding domain and the CRP-D1 effector domain, we have constructed ATF, and using the ATF libraries, we induced various phenotype changes in E. coli. Briefly, I would describe the strategies for the phenotype engineering with ATF libraries. By randomly assembling of different types of zinc finger proteins, we have constructed DNA binding domain libraries that consist of the 3 zinc fingers. And then they were fused to a CRP effector domain to create ATF libraries. And then with the introduction of ATF libraries to induce global perturbations of gene expressions, we induced various phenotype changes in E. coli, and then selected for the desired phenotypes, such as industrially important traits. First, I will mention the result concerning the cells with the osmotic tolerance phenotypes. Among the ATF transformers, we can select the cells resistant to osmotic pressure that inhibit the growth of the wild-type cells. And using the same approach, we can select the cold-tolerant cells induced by ATF. As you can see from this figure, the wild-type cells stop their growth at 15 degrees Celsius, but cold-tolerant cells induced by ATF grew well at 15 degrees Celsius. Finally, we can select the ATF transformers after heat shock for 2 hours at 55 degrees Celsius, a condition that severely inhibits the growth of wild-type cells. Among the survived ATF transformers after heat shock, we examined the growth profile of the best growing T2 thermotolerant cells. The T2 thermotolerant cells can grow well at high temperatures of 50 degrees Celsius. Next, to further analyze gene expression changes by the T2 ATF in the cells, we performed the DNA microarray analysis. The T2 ATF generated global perturbations of gene expression in the cells. Specifically, 133 genes were upregulated by more than 4-fold compared to the control cells, and 69 genes were downregulated. Among those genes, CPXP, OMPW, and MALEP exhibited the greatest change in their expression. Also, the regulatory regions of CPXP, OMPW, MALEP have the putative T2 ATF binding sites. So, to study whether CPXP, OMPW, and MALEP are direct targets of the T2 ATF, we performed the DNA band subset. We found that while the T2 ATF can bind the regulatory regions of CPXP, OMPW, and MALEP, when the putative T2 ATF binding site was changed to the sequence that the T2 ATF bind with much less specificity, the T2 ATF cannot bind that sequence. Therefore, the CPXP, OMPW, and MALEP are direct targets of the T2 ATF. In addition, these genes are involved in the resistance to the environmental stresses. OMPW is an outer membrane protein, and CPXP is a chaperone. MALEP transcription regulator that controls the expression of many genes, including SLP and HDA. In the T2 cells, T2 ATF represses the transcription of the BAR-LAB, which in turn leads to increased expression of the genes, such as SLP and HDA. SLP is also outer-membrane protein, and HDA is a chaperone. So, to examine the functional relevance of these target genes to the thermotolerance phenotypes, we either overexpress or knocked out the CPXP and the OMPW genes. And we also knocked out the BAR-LAB operant. When the CPXP or OMPW were individually overexpressed or co-expressed in the wild-type flags, the resulting flags were more resistant to heat shocks than the wild-type control flags. But they were much less resistant than the T2 thermotolerant flags. So, taken together, it seems that the ability of ATF to induce simultaneous modification to the expression of many genes is critically important for the obtaining highly improved thermotolerance phenotype. In addition, to study whether the various levels of phenotype changes can be obtained by ATF, we use the different copy number plasmids for the expression of ATF. As you can see from this figure, ATF can generate the different degree of thermotolerance phenotypes when it expressed at various levels using the different copy number plasmids, such as high copy and low copy plasmid. Indeed, in the transcriptional profiling, a total of 202 genes were in the T2 transformant expressing T2 ATF on high copy plasmids were differentially expressed by more than fourfold compared to the control set. 107 genes were changed in the LT2 transformant expressing T2 ATF on low copy plasmid. So, this result suggests that the various levels of phenotype changes can be obtained by controlling the level of ATF expressions. In conclusion, for the phenotype engineering of microorganisms, we have developed a novel artificial transcription package to induce global genetic perturbations in the E. coli cells. And using these ATF, we induce various phenotype changes in E. coli, such as the industrially important traits. Finally, the genes associated with resistance to the heat shock were then characterized. For example, OMPW, outer membrane protein, and the CPXP, chaperone, can provide structural stability and maintenance of protein folding state for the thermotolerance phenotype. I'd like to give a special thanks to my advisor, Professor Kim, and my colleagues. Many thanks for your attention.
Thank you very much. Are there questions? I have a question. I may have missed this. How did you decide which zinc fingers to use?
From the zinc fingers from the human genome was selected depending on the 4 amino acids, because zinc finger protein is composed of 2 beta acids and 1 alpha helixes. 1 alpha helix is critically important for specify the specific DNA sequence. So we select depending on the 4 amino acids and alpha helixes.
Did you have any problems with expression in E. coli?
No.
Did you have to add zinc?
Yes.
Did you have to have zinc to the media?
No.
Okay. Whatever. Any other questions? Okay, thank you very much. That's great.